You just wrapped up mastering the art of preparing specimens for optical microscopes, right? But how do we actually get the perfect specimens to look at in the first place? Let's bridge that exact skill with the real-world, fast-paced clinical reality of processing patient samples. We're going to take those fundamental 6 I's of microbiology you learned about previously and completely level them up to a professional clinical standard. So, to really solve this clinical detective story, we have to tackle a massive question. How in the world do we get from a completely messy clinical swab, think about a throat culture, a urine sample, or even environmental stuff like soil, all the way to a perfectly isolated microbe that's actually ready for analysis? It's a precise journey of advanced culturing and identification, and honestly, there's zero room for error. It all kicks off with inoculation, and look, this is way more than just randomly swabbing a plate. It's the highly deliberate, systematic, mechanical introduction of a biological sample into a container of nutrient medium. In a real clinical setting, we're using highly specific delivery tools. We're talking sterile inoculating loops, rigid needles, absorbent swabs, or graduated syringes to safely grab and transfer those inocula from really sensitive sources like blood or cerebrospinal fluid. Once inoculated, our sample needs a home, which brings us to the medium, or media if we're talking plural. This is a highly specialized, solid, semi-solid, or liquid, nutrient-rich chemical formulation. It's literally the food and environment the microbe needs to survive. But throwing food at it just isn't enough. To guarantee reliable growth during incubation, lab techs have to strictly regulate the physical variables. You've got to lock in specific temperature ranges, balance atmospheric gases like oxygen and carbon dioxide, manage the moisture, and absolutely ensure total sterility of your tools so you don't introduce any wild environmental contamination. If you get all those conditions just right, you get a culture. This is the payoff. It's the actual, visible, macroscopic accumulation of microorganisms resulting from all that active replication over time. It's kind of amazing when you think about it. You're literally watching the invisible, microscopic world multiply until it's staring right back at you in a laboratory container. Now, when you're actually in the lab doing this, you're going to rely on 3 main isolation techniques. First up is the streak plate method. This uses a sterile loop that you actually have to reflame between quadrants on a solid agar plate. It physically thins out the cells so you get distinct macroscopic colonies right on the surface. Then you've got the loop dilution or pour plate method. Here, you use liquid agar tubes kept around 45 to 50 degrees Celsius for serial dilution, which gives you colonies growing both on top of and deeply embedded inside the solid agar matrix. And finally, the spread plate method. For this one, you use a pipetting device and a glass hockey stick rod over dry agar to spread things out, giving you a really uniform distribution of easily countable surface colonies. But why do we do all this? Well, the ultimate goal of every single one of those isolation techniques is to achieve an isolated colony. This is a discrete, visible mound of literally millions of identical microbial cells resting on or within the agar. And here's the kicker. They all originate entirely from the replication of just one single parent cell. This isolated colony is the absolute foundational building block for all diagnostic testing. Seriously, you cannot proceed reliably without it. So how exactly does one lonely cell explode into a population of millions? Well, step 1 is the mechanical separation of that individual cell onto the agar surface. Step 2, we provide plenty of space and a continuous flow of nutrients. Then step 3 is the biological engine turning on, binary fission. This causes exponential doubling every single generation cycle. And finally, step 4 is the payoff, the formation of that visible, localized, macroscopic mass. It's literally exponential math happening right before your eyes. Now, as future allied health professionals, you absolutely have to know the difference between culture types for accurate charting. Let's use an analogy. Think of a pure culture as a pristine, high-security greenhouse that strictly grows red roses. Nothing else is allowed inside. It's highly controlled, just one isolated species. Now contrast that with a mixed culture. A mixed culture is more like a carefully planned display room designed to grow red roses right next to yellow tulips. Both species are meant to be there, they're growing together on purpose, and both are easily identifiable. But what happens when aseptic technique completely fails? Imagine that pristine rose greenhouse again, but somebody throws a rock and breaks a window. Suddenly, wild dandelion seeds blow in from the highway and take root right next to your prized roses. Now you've got a compromised, contaminated culture. Those dandelions are contaminants, unwanted invaders like airborne mold spores or skin flora that totally wreck your plant environment. In a clinical lab, these infiltrators will completely ruin your patient's diagnostic results. Assuming you avoid those rogue dandelions and your technique is flawless, your next move is to create a subculture. This just means taking a very precise portion of cells from your perfect, isolated colony and transferring them into a brand new container of sterile media. Think of it as a backup file. This secondary culture lets you safely maintain and multiply your pure pathogens so you can run extensive tests without losing your original sample. All right, once we have that pure subculture, the real interrogation phase begins. We need to systematically gather high-yield diagnostic data to uncover the pathogen's vulnerabilities and its genetic signature. We run biochemical profiles to track unique metabolic reactions, sugar fermentation, and gas production using those little inverted derm tubes. We figure out drug sensitivity by culturing the pathogen with different antibiotic discs to map out zones of inhibition, basically seeing which drugs actually kill it. We also use advanced immunological assays like targeted antibody antigen kits. And, of course, we sequence the molecular data, running PCR or gel electrophoresis to isolate highly distinct DNA base sequences. To turn that giant pile of data into a definitive identification, we follow a really strict pathway. We combine morphological classification, so documenting the macroscopic architecture of the colony and the microscopic cellular shape, with chemical staining analysis, running differential protocols like the gram or acid-fast stains. Then we fold in metabolic testing data using comprehensive multi-well strips like the API 20E system, which rapidly profiles exactly what nutrients our unknown organism is eating. So, the million-dollar question: how do we synthesize all this evidence? We take all those compiled morphological, metabolic, and genetic clues, and we feed them straight into automated software programs and diagnostic flowcharts. This computerized data matching crunches all the variables and finally slaps a precise scientific identity onto the pathogen. Boom, we've officially solved the clinical detective story. Now that we have successfully isolated and identified our culprit, we're left with a really fascinating question. How do these isolated pathogens actually function at a cellular level? As you jump into exploring prokaryotic cell structures and microorganisms in your very next chapter, keep this in the back of your mind. How does the microscopic anatomy of these pathogens drive the exact biochemical reactions we just used to identify them? Thinking about that will completely change how you approach clinical microbiology moving forward.